This video from an enhanced microscopic capture is depicting a very important process in keeping us healthy. This footage, shot over 2 hours and compressed to a few seconds, shows a neutrophil ingesting a fungal spore. About 350,000 bacteria and also a variety of fungi like aspergillus and numerous dust mites sleep with us every day on our pillow. Also, researchers have found out that an average computer keyboard can contain from 6 to 420 colony forming units of microorganisms like Staphylococcus, Pseudomonas, Enterococcus, mold and yeast. Many of these pathogens are harmful to the body and may cause diseases from a simple flu to serious diseases like sepsis or pneumonia. But we are not always falling ill, obviously, because our body has its own defense mechanism, which is collectively called the immunity. In general, the body's immunity is divided as innate immunity or non-specific immunity and acquired immunity or specific immunity. Innate immunity includes the physical barriers such as our skin, mucous membranes, cilia in the respiratory tract, and chemical barriers like lysosomes in the tears, ammonia in sweat, salivary amylase, the hydrochloric acid in our stomach, and so on. Also, the symbiotic bacteria on the skin and inside our body is competing with the pathogenic organisms for space and nutrition. A more important aspect of the innate immunity are our phagocytic cells. As the innate immunity acts equally against all and any invading pathogens, it is also called non-specific immunity. The acquired immunity is more specific to the type of pathogens and is activated later. As it is specific, the acquired immunity acts more efficiently. Acquired immunity functions in two ways. Humoral immunity, which acts by the means of chemicals called antibodies produced by the B lymphocytes, and cell-mediated immunity, in which the T lymphocytes directly engage the foreign material. Our body acquires specific immunity in different ways. There are natural ways, like any infection we get, or the antibodies passed over from the mother transplacental before we are born. And there are artificial ways like vaccination. As the development of specialized cells and antibodies takes time, specific immunity gets into action only few days after we get an infection. These blood smear images show the white blood cells, basophils, neutrophils and eosinophils, which are called granulocytes because of the presence of granules in the cytoplasm. These granules are filled with different chemicals like histamine, heparin, platelet activating factor, which will help them to kill the invading organisms. Also, these cells are called polymorphonuclear cells because of the lobed appearance of their nucleus. Monocyte is an agranular mononucleated cell which involves in phagocytosis. In the blood, it is not an active phagocyte, but it migrates to tissues and enlarge to become a macrophage, which has high phagocytic action. The granulocytes and monocytes are part of the innate immunity or non-specific immunity, and their function is to destroy the invading pathogens or particles by phagocytosis and to initiate inflammatory response. Lymphocytes are cells involved in the specific or acquired immunity. They are mononucleated agranular cells. They are of two basic types, T lymphocytes and B lymphocytes. Apart from these blood cells, other specialized cells called mast cells and dendritic cells also take part in the immune response. Mast cells are developed from the myeloid stem cell and they migrate to tissues where they mature. They are filled with histamine and heparin granules and plays a key role in allergic and inflammatory reactions and also wound healing and pathogen destruction. Dendritic cells are also formed from the hematopoietic stem cells and monocytes. They are present in areas communicating with external environment like the skin, lining of the nose, stomach and the intestines. They function exclusively as antigen presenting cells, that is, to engulf pathogens, process them and bring them to macrophages and antibody producing lymphocytes. Any unwanted particle entering inside our body are dealt with initially by the process called phagocytosis. The two main phagocytes are tissue macrophages, which are mature monocytes, and the neutrophil, which is a polymorphonucleated granulocyte. The monocyte is a mononucleated non-granular cell which circulates the blood in the immature form and are powerful when they migrate to tissues where they mature and enlarges fivefold up to 80 micrometers. When called for action, they can devour up to 100 pathogens before they die. Apart from circulating cells, a reserve storage of monocytes are found in the spleen, which can migrate to an affected site within 8 to 12 hours, sensing an inflammatory reaction. The other main phagocytic cell is the neutrophil. They are the most abundant among circulating white blood cells. Neutrophils are highly motile and can pass through tissue spaces easier than other cells. Because of these peculiarities, they are the first to respond to inflammatory signals from an affected site. Apart from phagocyting pathogens in a rapid rate, neutrophils are found to produce something called neutrophil extracellular traps, web-like protein fibers which helps to trap and destroy the pathogens. The primary reaction to any damage to our cells, whether it be by an invading organism, toxin, injury or heat, is the inflammatory response. The inflammatory response is initiated by the chemicals released by cells in response to physical or chemical damage, and later by the cells of the immune system which acts in the area. Cell damage causes the release of cytokines and antimicrobial peptides. This attracts the phagocytes to the site. The movement of phagocytes towards the released chemicals is called chemotaxis. Some of the many tissue products released by the tissue damage are histamine, bradykinin, serotonin, prostaglandins. Also cytokines like interleukins are released. These chemicals bring upon several changes in the surrounding tissue. The blood is prevented from clotting easily. This facilitates more blood circulation and transport more cells to the area. Nearby capillaries are dilated and the permeability of the capillary walls are increased. The macrophages, which are attracted to the area, squeeze through capillary membranes. This type of movement is called diapedesis. Also, the chemical reaction will stimulate the movement of tissue macrophages and activation of more of them from the bone marrow and lymph organs. There will also be clotting of fluid in the interstitial tissue around the injured site, which limits the spread of toxins and damage. The released chemicals and the reactions will produce local effects like redness, swelling, pain and heat. These are called the signs of inflammation. Also, the prostaglandins in circulation can lead to fever if the inflammation is sustained. A cell or chemical entering the blood or mucous membrane or any other tissue should be recognized as a pathogen to initiate an immune response. Accurate identification and early action is very important for the safety against diseases. It's equally important that the immune system distinguishes the invaders from the body's own cells so that own cells will be spared and invading pathogens will be correctly targeted. This identification is performed by recognizing certain features in the invading cells. Usually, phagocytes recognize special molecular patterns which are not expressed on mammalian cells but are found among bacterial cell wall components, viral genetic material, etc. Phagocytes have several specialized receptors for identification of these foreign protein or polysaccharide patterns. Those receptors are called pathogen-associated molecular pattern recognizing receptors. One of the receptors is called toll-like receptors, which are transmembrane proteins found on the cell membrane of immune system cells. They recognize molecules like bacterial flagellum, glycolipids, double-stranded RNA, etc. There is also mannose receptors which recognize different polysaccharides like mannose which are found on microbial surfaces. Another is node-like receptor which recognize certain patterns of molecular arrangements on foreign cells like peptidoglycan arrangements. All these receptors are looking for epitopes and large molecular weight polymers, which are not usually expressed in human cells. There are also receptors called damage-associated molecular pattern receptors, which recognize molecules released from damaged cells. The pathogens or damaged cells are also recognized by phagocytes by a mechanism of our body called opsonization. Opsonization is a coating of pathogens by molecules called opsonins, which include antibodies, C-reactive proteins, and complement proteins. As the word literally means, opsonins season the pathogens and make them more tasty to eat for the macrophages. The process in which one cell eats another is called phagocytosis. In phagocytosis, the phagocytic cell reach and engulf an invading particle in an amoeboid motion. The engulfed particle becomes enclosed in a vesicle called phagolysosome. The phagolysosome detaches from the cell membrane and remains as an independent vacuole. The cells enclosed in the phagolysosome are destroyed in two ways. One is non-oxidative destruction, in which the cytoplasmic granules of the phagocyte attaches to the phagolysosome and inject enzymes like lysozymes into it. Other method is oxidative destruction, in which enzymes like NAPDH and myeloperoxidase converts oxygen into reactive oxygen species and oxidizes the pathogenic cells. This is called oxidative destruction. The production of peroxides like hydrogen peroxide is very important in destroying certain fungus and bacteria, and the failure to induce oxidative destruction is an immune deficiency called chronic granulomatous disease. If an engulfed particle cannot be completely digested by the phagocyte, it will lead to the formation of giant cells by the fusion of such phagocytes and cause symptoms as in tuberculosis and giant cell tumors of the bone. As we discussed earlier, phagocytosis is mostly done by neutrophils in the blood and monocytes and derived cells in tissues. Eosinophil is another of the polymorphonucleated granulocyte. The phagocytic action of eosinophil is weak, but it has an efficient action against parasites as it attaches to the parasites and destroys their coating by releasing hydrolytic enzymes and reactive oxygen species like peroxides. Eosinophils also attach to the tissues where an allergic reaction occurs in response to the interleukins produced by mast cells and basophils. Eosinophils are believed to release cytokines which improve the action of other immune cells, and also they play an important role in limiting the effects of allergic reaction by disintegrating the antigen-antibody complex. Another granulocyte is basophil, and it is also associated with allergy and inflammatory reaction. Basophil, along with mast cells, which are located in the tissues around capillaries, release inflammatory substances like heparin and histamine to initiate inflammatory response and chemotaxis. Also, the immunoglobulin E attaches to the basophil and mast cells. IgE is mainly involved in allergic reactions. When a particular antigen attaches to IgE molecule specific for it, the basophil or mast cell to which the immunoglobulin is attached ruptures and releases reactive chemicals to the tissue and blood, which stimulates an allergic response. We have already discussed that the monocytes migrate to the tissues from blood and become macrophages. The macrophages that enter the tissue will remain there as large tissue macrophages, ready to engulf invading pathogens. This specialized macrophage system in peripheral tissue is called the reticuloendothelial system or the monocyte macrophage cell system. These specialized phagocytic cells are different in each organ system, but they function similarly: ingest and neutralize invading cells. Examples of reticuloendothelial cells include Kupffer cells of the liver alveolar macrophages of the lungs, microglial cells in brain, mesangial cells of the kidneys, histiocytes of the skin, etc. These tissue macrophages can also break off and become mobile macrophages if the situation demands. The response of the innate immunity to an invading particle follows the following steps. At first, the tissue macrophages engulf the particle. The antigen from it is presented to the other immune cells and inflammatory chemicals are released. The chemical release attracts neutrophils to the area. The feedback with cytokines will simulate the release of stored neutrophils from the bone marrow to the blood. This increase in number of neutrophils in the blood is called neutrophilia. Up to 5-fold increase in neutrophils can happen in acute inflammations. Monocytes and neutrophils will invade the affected tissue. Then the cytokines and other complement proteins will stimulate the production of granulocytes and monocytes in the bone marrow. It will take up to 4 days till new cells are formed and are mature enough to be released. All these are constantly controlled by the feedback mechanism from the cells by cytokines, colony stimulating factors, etc. These chemicals in turn also activate the lymphocytes, which are responsible for further action with a specific immunity. Adaptive or specific immunity is acquired in various stages of life. At first, by the transfer of antigens from mother to fetus, and later as a response to infections or through vaccination. It's divided as humoral immunity, which is mediated by chemicals called antibodies produced by B lymphocytes, and cell-mediated immunity performed by T lymphocytes. As we discussed in the previous part, the adaptive immunity takes days to develop after a pathogen invasion, but it is very powerful owing to its specificity. Also, cells of the adaptive immunity form memory cells which will act faster and stronger if the same pathogen attacks us a second time. As you see in this graph, the antibody concentration as a response to the secondary attack of the same pathogen is starting earlier, is higher, and lasts longer. All lymphocytes develop from the same stem cell in the bone marrow but are functional only after a processing. This processing differentiates the lymphocytes and enables them to perform their specific function. Some of the lymphoid progenitor cells migrate to the thymus where they are processed into cells which can directly kill pathogenic cells. They are called T lymphocytes. T lymphocytes are further specialized as helper T cells, cytotoxic or killer T cells, and suppressor or regulatory T cells. Helper T cells are also called CD4 plus cells, and cytotoxic T cells are also called CD8 plus cells due to the presence of corresponding receptors on their surface. The common lymphoid progenitor cells which enter the thymus undergo regulation of their genetic expression and become a double positive cell which at first express both CD4 and CD8 receptors. These cells move to the thymic cortex where they are presented with self-antigens. Only the cells which react strong enough to the self-antigens will get a survival signal, and the rest of the cells with weaker reaction to the self-antigens will die off. The type of receptors by which they attach to the antigen determines whether they become a helper T cell or cytotoxic T cell. The cells which underwent the above positive selection will migrate to the corticomedullary junction of the thymus, where the autoimmune regulatory gene presents the cells with self-antigens from different tissues of the body. Those cells which react strongly to the self-antigens get a signal for destruction. This is called negative selection. About 98% of lymphocytes are destroyed in the thymus by positive or negative selection, and 2% leaves the thymus as naive T cells. These naive cells are later activated when presented with antigens by antigen presenting cells like macrophages or dendritic cells. Next type of lymphocytes are the B lymphocytes, which are processed in the liver and bone marrow before birth and in the bone marrow after birth. The B comes from bursa of Fabricius, which is a hematopoietic organ in birds, where the cells were first identified. Like T lymphocytes, B lymphocytes also undergo a positive selection at first, which is antigen independent, and then a negative selection with exposure to self-antigens. The activation of B lymphocytes for immune function mostly involve helper T cells and is called T cell dependent activation. The activated T cells express a protein ligand called the CD40L on its surface, which binds to its receptor in the B cell called CD40. This, along with the cytokines called interleukins released by the T cells, activate the B cells which undergo proliferation. If the antigen is met by the B cell for the very first time, then a reaction called somatic hypermutation takes place in the antibody, where substitutions for molecules takes place in the variable portion of the antibody to make it adapted to the new antigen. Activated B lymphocytes can also switch one type of antibody to another if necessary by a reaction called antibody class switching, in which, for example, IgG can be switched to IgD or M, in which the constant portion of the heavy chain in the antibody is changed. These antibodies are later released into the circulation. The activated B cells mature and develop to plasma cells to release antibodies to their full potential. In their full potential, the antibodies are released at a rate of about 2000 molecules per second per cell. Some of the cells are stored as memory cells before becoming plasma cells. These memory cells will be activated faster when the body is exposed to the same antigen for the second time. This selection process of lymphocytes are very important to protect the body. It's during this selection process lymphocytes familiarize with the antigens of our own body. Any error in proper selection will lead to the lymphocytes destroying our own tissue and causing the condition called autoimmunity. One of the vital processes in the immune reaction is antigen presentation. Antigen presentation is the mechanism of processing a pathogen, a tumor cell or toxin by phagocytes or lymphocytes and presenting it to T cells. This process is done mainly by macrophages and dendritic cells, but the B lymphocytes can also act as antigen presenting cells. When a pathogen enters the body, macrophages or specialized antigen presenting cells engulf them and disintegrate it. A portion of the foreign material called epitope is processed and presented on the surface of the antigen presenting cell to be identified by T lymphocytes. The antigen presentation is done by a protein called major histocompatibility complex, or the MHC. The specialized protein molecules are found in all vertebrate animals and are coded by genes of the same name. In humans, the genes are called major histocompatibility complex genes, and sometimes the proteins are referred to by the name human leukocyte antigen, or HLA. There are mainly two types of MHC proteins, class 1 and class 2. MHC class 1 molecules are present on the surface of all nucleated cells of an organism. As these molecules are coded by a number of genes with a large number of alleles, two humans will rarely have the same MHC class 1 proteins. This is an important factor in the rejection of transplants, because the T cells will recognize MHC proteins from a different person as an antigen and will try to destroy those cells. The term histocompatibility, meaning the compatibility of tissues, is derived in this context. If a cell is diseased and the normal protein synthesis or gene transcription of a cell is impaired, either because of a viral attack or because of expression of a cancerous gene, a byproduct of the new impaired protein is transferred to the cell surface and is expressed on the MHC class 1 receptor. This protein on the MHC class 1 is identified by cytotoxic T cells, which kills the disease cell. So MHC class 1 protein is presenting the antigen to cytotoxic T cells. Some viruses and cancers escape this mechanism by downregulation of the MHC genes. That's how they become more dangerous. MHC class 2 molecules are expressed only by the specialized antigen-presenting cells like macrophages or dendritic cells. They digest the circulating pathogens, and a peptide of the material is transferred to the surface and expressed on the MHC class II protein, which can be recognized by helper T cells, which is in turn activated to release cytokines to trigger response from other immune cells. MHC class III genes are relatively newly identified and are not antigen binding but believed to produce molecules of the complement system and also initiate inflammatory response. As mentioned before, antibodies are specialized molecules produced by B lymphocytes in response to activation by an antigen. Antibodies are produced specifically according to the infective antigens and usually takes about 3 to 5 days to be released if the body is encountering the antigen for the first time. The response time of the second exposure is lesser, about 2 days. Antibodies are soluble glycoprotein molecules classified as gamma globulin and are immunoglobulins. Structurally, they are pairs of light and heavy chain polypeptides. Most commonly, there are 2 light chain and 2 heavy chains, but the immunoglobulin M has 10 heavy and 10 light chains, while IgA has got 2 pairs each. All antibodies have a constant part which determines its physical properties like diffusibility, attachment to cell membrane, etc., and a variable part which determines the specificity of the antibody. As we discussed earlier, when a B lymphocyte is activated for the first time, it can change the gene encoding to change the variable portion of the antibody to attach to a particular antigen. This process is called somatic hypermutation. Also, the B lymphocytes can change the type of antibody it is producing according to the type of stimulus received. For example, from IgG to IgM by a process called antibody class switching. There are 5 types of antibodies identified in the human body. The most abundant is IgG, which is about 75% of all the antibodies. In humans, IgG has 4 subclasses. Fetal immunity is largely dependent on IgG as it is the immunoglobulin which is transported across the placenta to the fetus. It's also the first antibody to be produced by the B lymphocyte along with IgM and appears in the blood about 3 to 5 days after an infection. It is found as circulating in the blood and also in other extracellular fluids and saliva in a healthy person. IgM is also a primary response antibody. Even though its relative volume is small, because of its structure, it has more antigen binding sites and is very effective. IgM helps in agglutinating pathogens and render them less mobile and easier to be destroyed. IgE is responsible for allergic reactions as it binds to mast cells, eosinophils and basophils and can activate these cells to release histamines, heparins and other cytokines when encountered with an allergen. This antibody is also found in higher concentration in people with parasitic infections. Immunoglobulin A is mostly found on mucosal surfaces and acts as a defense against inhaled or ingested pathogens. It is also found in secretions like tears, saliva, and is a predominant antibody in breast milk. It is highly effective in neutralization of toxins. It has 2 subclasses, A1 found mostly in serum and A2 found mostly in colostrum, breast milk, saliva, etc. IgD is the least understood antibody, but it is believed to be associated with mast cell and basophil activation. Antibodies function in multiple ways to protect the body. They attach to specific antigens presented on the pathogens. These attachments make the pathogens stick together to form clumps and they are rendered immobile and more susceptible to phagocytosis. This is called agglutination. Some soluble toxins like tetanus toxin is made inactive by the antibody attachment because they become insoluble and precipitate. In other cases, the reactive sites of a pathogen or a toxin is covered by the reactive site of an antibody and it is neutralized. Antibody attachment can also destruct the cell wall or the structure of the pathogen and cause lysis. The antibody which is bound to antigen molecule or the antigen antibody complex also activates a protein called complement 1, which results in a chain of reaction further enhance the immune response. This is called the complement system or the complement cascade of reaction. The complement system contains about 30 proteins which are synthesized in the liver. They circulate in the blood in an inactive form. They are activated when an immune response happens. The first method in which they are activated is called the classical pathway of the complement system. In the classical pathway, the antigen-antibody complex binds to a protein called complement 1 or C1. This activates the C1 molecule, which splits another molecule C4 and C2, and the reaction continues. The complement pathway can also be initiated directly by a particle from the pathogen surface when it attaches to a protein called mannose binding lectin. This pathway is called lectin pathway or the MBL pathway. Sometimes the complement system protein C3 is activated directly by a pathogen surface without antigen binding. This is called the alternate pathway of complement system. Once activated, the complement proteins increase the inflammatory response. These proteins also act as opsonins to improve phagocytosis. The N molecules of the cascade can also cause lysis of pathogen cells. Other effects are agglutination of the infectious material, neutralization of toxins, enhancing chemotaxis, and mast cell activation. To sum up, when the primary defense of the body, like the skin, is breached, active immunity is put into action. Tissue phagocytes engulf and destroys the invading pathogen. An inflammatory response is also initiated and chemical signals are sent calling for help. More phagocytic cells like neutrophils and monocytes are called for action in the area. A portion of the infective agent is presented to specific immune cells by antigen presenting cells, and specifically acting B lymphocytes and antibodies are put into action. Interleukins and complement proteins improve the function of immune cells. More production and differentiation of immune cells takes place in bone marrow and lymphatic system. Enhanced phagocytic action and destruction of cells by effector T cells takes place. Neutralization of toxins happen due to the action of antibodies.